Hello friends, today we are going to discuss about red blood cells. In this video we will discuss on morphology of red blood cell, and secondly we will discuss Hello friends, in this video we are going to discuss about two things. First we will discuss morphology of RBC and its benefits. The second we will discuss function of RBC, and the third will be metabolism inside the RBC. And in our next video we will discuss about erythropoiesis. So in this video now we are going to start with morphology of RBC. So as we know that RBC is your biconcave shape, like this. RBC is your biconcave shape cell, okay, and at center it is 1 micrometer in thickness, and at periphery it is 2.2 micrometer in thickness. So RBC is a biconcave cell, non-nucleated cell, means nucleus is absent in RBC. Okay, now coming to the benefits, why RBC has biconcave cell, what benefits RBC is getting because of biconcave cell shape. So the first one is that due to biconcave shape, surface area is increased in your RBC. Okay, surface area is greater in RBC. So if surface area is increasing, it will increase diffusion of gases. Okay, because of large surface area there is large diffusion of gas. Okay, so diffusion of gas becomes easy because surface area is more in RBC due to biconcave shape. The second benefit is this RBC can withstand in hypotonic solution. If RBC is placed in your hypotonic solution, okay, hypotonic solution, suppose this RBC is put up in your hypotonic solution. This is a container in which hypotonic solution is put up. So in hypotonic solution what will occur? H2O from a hypotonic solution will enter inside your RBC. Okay, H2O will enter inside RBC, and RBC slowly first become spherical. Okay, so RBC first become spherical, and then RBC undergo bursting. If suppose the shape of RBC is spherical, suppose if shape of RBC is spherical, so shape of RBC is if spherical, then water will enter inside the RBC, and RBC quickly burst if it is placed in hypotonic solution. But the shape of RBC is biconcave, so at first water from hypotonic solution will enter inside the RBC, and RBC from biconcave cell will convert it into a spherical cell, and then it will undergo bursting. So RBC has time, okay, has time lag between after putting up in hypotonic solution and bursting. So we can prevent bursting of RBC, because RBC take more time in bursting because it has to convert it into a spherical, and then it will go for bursting, while a normal spherical cell will burst as we put that cell into hypotonic solution. So this is the second benefit of biconcave cell. The third benefit of biconcave cell is that it can squeeze through capillary very easily. Okay, squeeze through capillary very easily because of this shape. Because of this shape it can squeeze capillary easily, because suppose it is a spherical shape, then it's, so suppose this is your capillary, from there it has to skew, so this can easily pass out through this pore. Okay, but this spherical shape cell cannot easily pass out through this pore. So this biconcave cell also helps RBC squeezing throughout the capillary very easy. Okay, so these are the three benefits from a structural point of view. First, that is biconcave cell helps to increase surface area from the RBC, that will increase your diffusion of gases. The second, RBC can withstand with hypotonic solution more than a normal cell because of biconcave cell, because biconcave cell first will convert it into a spherical cell, then it will go for bursting, and the third is your squeezing through capillary becomes easy in biconcave cell due to biconcave shape of the cell. Okay, now so these are the points about the morphology of RBC. Now we are going for second part. One more thing we are going to discuss here, one graph, that is your Price Jones curve. Okay, we are going to discuss about Price Jones curve. Basically this graph is between diameter of RBC and number of RBC. Okay, this graph is between number of RBC and diameter of RBC, and it is found that this graph has peaked at about 7.1 micrometer. Okay, this graph has peaked at about 7.2 micrometer. Clear? Now it means the average diameter of RBC, the maximum number of RBC having diameter of 7.2 micrometer. Clear? So this is the peak, okay, number of RBC is maximum here. So the maximum number of RBC has diameter of about 7.1 micrometer or 7.2 micrometer. This is the Price Jones curve, and according to this curve we can easily state the condition of RBC, that is in the condition of microcystic anemia and macrocystic anemia. In microcystic, the RBC size get decreased, and in microcystic the size of RBC increases. So we can, based on this Price Jones curve, we can easily say microcystic and macrocystic anemia. How? If this graph shows if this graph shows rightward deviation, okay, it means the average, sorry, this is the graph, it means it is showing that the average diameter, okay, average diameter had been increased. Suppose this point is your 7.6 or 7.7, okay. If the graph is shifting towards right, then it will give us idea of macrocytic anemia, okay. And if this graph is shifting towards left, that is like this, okay, so this will be the condition for microcytic anemia, okay. So microcytic and macrocytic condition of RBC can be easily analyzed by your Price Jones curve. Clear? So right shift will indicate about macrocytic and your left shift will indicate about microcytic nature of the RBC, okay. Where when the graph shifts toward the left, it means RBC is a small in size in comparison to normal, that is 7.1 micrometer. Clear? So this is the basic use of this Price Jones curve. So this is all about your morphology of the RBC. Now we are going to discuss about the metabolism inside RBC. Now, as RBC has no nucleus, okay, no endoplasmic reticulum, no ribosome, okay, so many metabolism which are occurring in normal cell is absent in RBC. But still, RBC required ATP because it has to run the sodium potassium ATPase pump to maintain the ionic environment inside the cell. So RBC require ATP, and this ATP requirement is fulfilled by your glycolysis process, that is your EMP pathway, okay. Nucleus, ER, ribosomes are absent, so in RBC many processes which are occurring in normal cell will not occur here. But still RBC has to maintain ionic environment, so sodium potassium ATPase must be running there, and for sodium potassium ATPase ATP is required. So ATP is delivered by your glycolysis process which is running inside your RBC, okay. So this is the first process which is going inside RBC, that is the glycolysis. And through glycolysis, okay, through the glycolysis 2 ATP gain is there, okay, plus 2 NADH. This 2 ATP is utilized. Now second is your HMP pathway. The second is your HMP pathway. Through this HMP pathway in RBC, NADPH is synthesized, okay, NADPH is synthesized, which is reducing equivalent, and this NADPH, okay, this NADPH is synthesized by the enzyme, that is your dehydrogenase enzyme, okay. And this is a glucose 6-phosphate dehydrogenase enzyme, the first enzyme in the HMP pathway, okay. So HMP pathway is running in your RBC, which is synthesizing NADPH from glucose 6-phosphate dehydrogenase enzyme help, and this NADPH is reducing your oxidized glutathione, okay. Oxidized glutathione is reduced into reduced glutathione, because this NADPH is supplying hydrogen, and this hydrogen is combining with this oxidized glutathione, and we know addition of hydrogen will lead to reduction of something. So this oxidized glutathione, after combining with hydrogen, converted into reduced glutathione. Why this glutathione has been oxidized? So coming to this point, if RBC is exposed to your oxidant molecules, okay, which can oxidize RBC, so preventing RBC from oxidation, glutathione itself get oxidized. So in presence of oxidant molecules, oxidant environment, glutathione will undergo oxidation and become oxidized glutathione, okay. And this oxidized glutathione must be converted into reduced glutathione, so that it again will be converted into oxidized glutathione when RBC is exposed to your oxidant molecules, okay, oxidizing molecule. So NADPH must be there so that it can convert oxidized glutathione into your reduced glutathione, so that this cycle can be continued. So this is oxidized glutathione is converted into your, this is the glutathione, normal glutathione is converted into oxidized glutathione whenever any exposure of oxidizing molecules, okay. And this oxidized glutathione in presence of NADPH is converted into again reduced glutathione, which again, if given exposure to these oxidant molecules, again be converted to oxidized glutathione, so that the cycle can be run. If NADPH is deficient, means any glucose 6-phosphate dehydrogenase is deficient in RBC, then RBC membrane can be oxidized, because there once glutathione is converted into oxidized glutathione will not be converted back to reduced glutathione because of the deficiency of glucose 6-phosphate dehydrogenase, the deficiency of NADPH. So this process is helping to maintain the RBC membrane fragility. Clear? Now So these are the two basic processes, metabolic processes that are running inside your RBC. Clear? These are the two basic processes which are running inside the RBC. Now last portion of the discussion is our functions of RBC. Functions of RBC. So the first basic function of RBC is your transport of gases, as we all know, transport of gases. Okay. The second, as RBC contains hemoglobin protein, as you all know, RBC contains hemoglobin protein, and protein always take parts in your buffer. So hemoglobin, due to presence of hemoglobin protein, RBC also help in buffer. And this hemoglobin buffering is due to imidazole group of your histidine residues. Okay, the buffering of hemoglobin protein is due to imidazole group of histidine residues. Clear? We will discuss in detail when we will discuss about buffers. So first is your transport of gases, the second is your hemoglobin protein, which has a role in your buffering, and the third is your maintaining viscosity of blood. RBC is maintaining your viscosity of blood. Clear? Now these are the three basic functions of the RBC. Now one question arises, let's suppose if hemoglobin is present outside the RBC, hemoglobin is present outside the RBC, if hemoglobin is not inside the RBC, then what will happen? So the first point will be, if hemoglobin is present outside the RBC, then this is a solid particle, so it will also increase osmolarity of blood. Okay, first point is it will increase osmolarity of blood. The second will be it also increases viscosity of blood. Okay, it will increase viscosity of blood because it is a solid particle. Okay, so it has two impacts on blood. First, it will increase osmolarity of blood, and it will increase viscosity of blood. And if these two are increases, then heart has to be increase its cardiac output, heart rate, because the flow of blood is slowing down. Okay, which will lead to stagnant hypoxia. So heart has to overcome these problems by increasing its heart rate and cardiac output. Clear? Now we are also one point that hemoglobin, if hemoglobin comes out of the RBC, hemoglobin may be filtered through your kidney. This may be one more reason. Now coming to the last part, that is the clinical applied part of the morphology of the RBC, that is your hereditary spherocytosis. Hereditary spherocytosis. So this is basically a autosomal dominant disease. Okay, this is a autosomal dominant disease. In this RBC, which is normally a biconcave cell, converts into a spherical cell. Okay, and this is mainly due to deficiency of two proteins, either deficiency of two proteins or defective in the formation of these two proteins. That is your first one is ankyrin. Okay, first one is ankyrin protein, and the second one is your spectrin protein, because these two proteins are necessary for the maintaining biconcave shape of the RBC cell. If these two proteins are defective in nature or deficient in RBC, then these two proteins, ankyrin and spectrin, are responsible for your maintenance of biconcave shape of RBC. And if these two proteins are not synthesized in a proper manner, okay, or defective synthesis, then biconcave shape of RBC will not be maintained, and RBC will be converted into a spherical shape. And if RBC becomes spherical in nature, then two things will occur. Its osmotic fragility will increase because it cannot withstand hypotonic solution very much. Okay, if the shape was of biconcave, then we have discussed that biconcave shape cell can withstand your hypotonic solution more than a spherical cell, because biconcave cell first converted into a spherical cell, then it will undergo bursting, then it is getting more time. Okay, but a spherical cell, after putting up in your hypotonic solution, it will undergo bursting. So hereditary spherocytosis will increase your osmotic fragility. Okay, so this is all about your morphology of RBC, functions of RBC, and metabolism which is occurring in RBC. In next video we will discuss erythropoiesis. So thank you.